protein turnover ataupun turnover protein. Okay. Okay, boleh dengar saya? Can you hear me?
Yes.
Boleh ya? Okay. So, alright. So if you tengok, okay, if you look at the this tajuk, the title of the manuscript or the journal, yeah, protein turnover in aging and longevity. Lepas tu, apalah, you okay, take a look the abstract. Okay, I want everyone. I give you 5 minutes, okay, to read this abstract here. Okay, this abstract here. Okay. Kenapalah saya suruh baca? Okay. Alright. So the concept the protein turnover. So protein turnover ni, because the process of protein turnover, so is actually a process where it determine the half life, the half life of the certain protein. So bila dikaitkan dengan aging and longevity, so this is just a part of the study saying that if there is the end of the, or there is no longer production of the certain protein or certain enzyme produced in a certain level of certain stage in someone's life. So then it is going to start with the aging, aging. And if someone, they can maintain the formation of production of a certain enzyme or protein, during the for certain metabolisms, then it may contribute to the longevity as well. So somehow it depends on the protein tersebut. Okay, how is the metabolism of the protein, then it can actually contribute whether to the aging or longevity. So that is the how that is a very big contribution, very big contributions of a major role of the protein. But I'm saying that this is just a part of the the idea about the protein turnover. Okay, so let's take a look next. Okay, so here we're going to look at the principle of how the cellular proteins turn over. Okay, how we're going to relate this to the why it is said that the protein turnover is actually determining the half-life of the protein. Yeah, so then we're going to look at why the cellular proteins differ, okay, in their half-lives, because certain proteins or certain enzymes, they have like about 15 days or 2 weeks or 1 month, or even there is a certain protein or enzyme, they have just a half-life of 2 seconds, just a few seconds. So why is that? why they have such a different half-life. Okay, and in the same time also in this topic uh, you hopefully that you will be able to describe the mechanism of cellular protein degradation, okay, um, uh, via this ubiquitin proteasome pathway. Okay. Okay, so the proteins that we're talking about, they have what we call as the predetermined lifetimes. Okay, predetermined lifetime or half-life. And if there is dah sampai masa, ketika dah sampai masa, dia sampai dia punya half-life tu, so it will be replaced in the regular basis. So that is the thing with the proteins. And the proteins are constantly, okay, it will be constantly synthesized. It will be it will be keep replacing in the regular basis to make sure that this amino acid pool is uh, accumulate as usual, okay, function as usual, and those the protein that no longer needed, it should be degraded back. Okay, should be degraded. Okay, so this is the proteins, and so this is the functional proteins lah. Okay, it's the functional proteins that finally active. Okay, but then it's actually derived or comes from these amino acid blocks. Okay, amino acid blocks, and then this amino acid is go through what we call as process of protein synthesis. So process protein synthesis, there are many stages. Okay, there are many stages for protein synthesis, but we are not going to talk about the stages that takes place in protein synthesis, because we have one specific topic for this, just to discuss on the protein synthesis. Okay, so here then we get the protein, active protein, and the protein, when it comes to the half life of the protein, then it will be degraded. Okay, it will be degraded and will be converted or changed into a simpler amino acid pack. And this is where this amino acid um, this is the amino acid pool, yeah, the amino acid pool. And somehow the amino acid is just not to contribute to photosynthesis, but of course another metabolism as well. And okay, so the same, I think this is the same concept that I've showed you just now in the previous slides, in previous topic, about how the amino acid pools contribute to the formation of protein body, yeah, and how the part of the amino group of the amino acid will be synthesized to the formation of the nitrogen compounds, and those amino acids that no longer needed, then it will be catabolized or will be degraded, and the parts of the amino group of this amino acid will be excreted as the urea and carbon dioxide. Right? And of course, the source of the amino acid pool here is actually comes from the dietary amino acids, because of course we are not the apa, autotroph. Okay, we were not the autotroph, the one that can create the energy ourselves, like the plants. Yeah, so we are actually the heterotroph. So we have to eat something. We have to eat the source of the plants or animals. Then we can get the dietary amino acids. Okay. So um, so this is the overall idea about the protein turnover, because um, uh, of course a protein, they involve in a certain pathway. So this continuous synthesis and breakdown of the body proteins is really important, to make sure the maintenance of the optimum functional protein, because we know when certain protein or certain enzymes, they have a different half life, okay, different half life. So they need to keep replacing in the regular basis. We have to maintain. Yeah? We have to maintain the existence, right? Existence of this protein of this amount of the protein with the correct concentrations to make sure it functions very well. And this is what we call as a dynamic process. Okay, a dynamic process we have where this maintenance of the synthesis and the breakdown of the protein should be tightly regulated. And if let's say something happened to the protein behavior, which is it behave abnormally, then it will be then uh, yeah, there will be something else happen, which is the abnormal cell behavior will contribute or will cause a certain disease or yeah, some abnormal functions of the cells. All right. Before we go into details to see how the protein turnover happen. So this is an overview of how the amino acids catabolism takes place in mammals. Yeah, so it started with the whatever the dietary protein or intracellular protein, and it will degrade it into the amino acids. And yeah, I've already mentioned about this just now, where the amino acids, because we know the structure formula of amino acids, the basic amino acid, the basic structure. If you refer back to the first lecture that I showed you about the structure of the amino acids. So it contained the amino group, right? The amino group, the carboxyl group, and also the hydrogen side chain. Okay, the side of the side, the side chain of the amino acids. So the amino group in the amino acids, this is the one that will be contribute to the formation of the ammonia. Okay, and then it will enter the urea cycles. And those, the rest, except the amino group, it will be degraded into as carbon skeletons, and it will enter the TCA cycle towards the oxaloacetate and to produce the glucose, yeah, in the process of gluconeogenesis. Okay, so that is the idea about the major role. You can see the major role of the protein in the metabolisms. Okay, and of course, when the protein when we said that the protein has to be accumulated and sometimes it has to be degraded. So there will be a different states of formation of the protein turnover. So it depends on how the protein is accumulated or it will be degraded. So this will contribute to this formation, what we call as steady state or full expansion or full contractions of the amino acids. So this is a steady state where the synthesis of the amino acids is similar to the ones that degraded. Okay, and of course the transcriptome, uh, the formation of the transcriptome is ideal with the one that occur in this part, the metabolome, okay, the production of the metabolites. And this is the pull expansion. Okay, the pull expansions meaning that there will be, of course, there will be when if you take a look at the expansion, okay, the expansion is something that expands, yeah, something that expands. So when it relates to the pull expansion itself, so it must be the increase, yeah, the increase of the synthesis, yeah, the increase of the synthesis, and then there will be unchanged in the part of the degradation. So that's why there will be the full expansion. Or there or in another situations, there will be unchange of the synthesis. Okay, unchange of the synthesis. No, nothing is synthesized. Nothing new is synthesized. But then there will be decrease in the degradations. This also will be contribute to the full expansion. Okay, and and another state is the pull contractions where so there are also two states which contribute to the pull contraction of the amino acids. So the first one is where the amino acid is the synthesis of amino acids unchanged, yeah, unchanged, and then but then the the there will be increase in the degradation. Okay, there will be increase in the degradation. So and the same thing goes to here. Okay, there will be decrease in the synthesis and also unchanged degradation. So these two will create the pole contraction. Yeah. Okay. Um, so this is related to how the protein we get the negative nitrogen balance. Um, so we have discussed about nitrogen negative nitrogen balance, right, in the previous topic, the previous topic, where we have the positive nitrogen balance and negative nitrogen balance, and we have the balance, we have the exactly nitrogen balance. But here there is a certain condition, a certain variations that actually contribute to the what we call as negative nitrogen balance. Some but the idea is that the degradation, yeah, the degradation of the protein, the degradation of the protein will be higher. Okay, will be higher compared to the ones that is synthesized, and it will really contribute to the negative nitrogen balance. Okay. So all right. So before we continue, I'm going to show you these slides here. Okay, this video. So hopefully everybody wake up lah. I know petang-petang ni semua orang mengantuk and kelas dengan my was macam ni. Okay, so please listen to this.
Sorry, doctor, we can hear the sound.
Oh, okay, sorry. Okay, I need to. Okay, I cuba dulu ya. Kalau tak dengar juga, let me know.
Okay.
Dengar tak?
Oh, tak dengar.
Tak dengar juga. Okay, I will try another one. Tak dengar juga.
Ya,
boleh dengar tak?
Tak boleh.
Tak juga. Tak boleh. Okay, okay. Then I need to
Your eyes and your body see the food you eat in very different terms. For example, if you have a cheeseburger sitting on your dinner plate, your eyes see a bun, a hamburger patty, and a slice of cheese. Your body is a supply of nutrients that it can break down and reassemble to care about your body. The cheeseburger fat in this body.
boleh.
In this lesson, we will focus on how your body much like your kitchen.
Doktor,
Yeah. Because protein
boleh pasangkan caption tak sebab macam bunyi dia tak berapa dengar.
Oh ya, okay, kejap. Sekejap I ulang balik.
Thank you, doktor.
Okay, I start balik lah. Okay. Very good. If you have your eyes on hamburger and cheese, your body gives us a lot of nutrients that break down. It's in order to focus on how your body makes the balance. See this your body with the protein is pretty important. The amount of protein you need in your diet is based on your pounds. Now remember that most of the nitrogen we consume comes from those nitrogen acids in the protein cell. The nitrogen is free because the body. When you get nitrogen, you're eating the right. Your body always likes to strength. So when you're nitrogen, nitrogen orchestrates this shit. It just individuals that do not have adequate amounts of protein in foods available to them, as we might see in people with undeveloped countries or in dieters who reduce their it can also happen. For example, that carbohydrates are protein
Alright, so that's it about the protein turnover. Yeah? Protein turnover to relate to the um, balance, nitrogen balance. So there are three states, three states of nitrogen balance. So that is the first one is of course the nitrogen balance, and then the second one we have the positive, positive nitrogen balance, and then we have the negative nitrogen balance. So there are the different factors that contribute to this formation of these three states. Yeah. So okay. Okay, so this is the formation of the amino acid pool. Okay, the formation of the amino acid pool. So basically, the average for an adult to take the source of the protein is 100 gram. Okay, 100 gram in a day. So what does this amino acids will do with its amino acids? So we have the part where this amino acid will synthesize. Okay, we synthesize the purines, pyrimidine, neurotransmitters, glucose, and so on. So that's for the formation of the ATP or energy, and then some of it will go to cellular protein, active cellular protein, and then some of it also will be break down back to these amino acids, amino acid pool, and some of these amino acid pool also will lost, will lost in a form of urea or urine. Okay, so about 35 to 55 gram per day, and so and in a certain condition, we should also understand that not all these amino acids will just go to one way towards this way or towards the formation of cellular protein, but of course the dietary protein also will, in the form of the protein, also will be converted to the glycogen or fat, and will be stored as well. Okay. Okay, let's take a look at the mechanism of the protein breakdown, because since we know that certain proteins, there will be, they have a certain different half-life. So how the protein is going to break down. And okay, so why do cellular proteins turn over? Why do proteins have this kind of turnover? Why don't just the proteins it will when it produced, and then we keep maintaining use the proteins all times, until the end of our life. So but then there is no such thing. So these cellular proteins always keep turn over, and keep replacing in the regular basis. So this is because the proteins, it can be damaged, and the proteins can be damaged. Protein ni macam juga cell. Okay, so dia boleh rosak juga because due to this oxidative damage and need to be destroyed. So tak boleh ada lama-lama dalam badan, so it has to be destroyed. So what is the oxidative damage? Anyone can explain to me what is the oxidative damage here? Why such oxidative damage will cause something back to the protein? You have no idea. Nobody. Tak ada siapa yang. What is the oxidative damage? Tak tahu. Okay. Please find out what is the oxidative damage. Okay, so the second one is of course, as protein. Okay, protein, because it will go through the process of translation, transcription dan sebagainya. So during along the process, not all the proteins will be correctly produced. Okay, it will be some errors in this amino acid sequence, and it should be removed lah. Okay, because this if this protein is going to enter the bloodstream or the cells, then it's going to be something abnormal happen. Yeah, so it should be removed. Okay, so that's why we have the proteins and over. And then the next one is the used proteins cannot be stored. Okay, some proteins or some enzyme, when we can just use it once. Okay, once, just only macam pakai buang, pakai sekali saja, and then we should remove it because this amino acid cannot be recycled. or 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 it can be recycled. It should be it should be degraded into a simpler amino acid, and then kita boleh recycle lagi sekali. And then the last one is the regulation of the hormone and enzyme levels. So it depends on the regulation of the hormone and enzyme. So some of the some of the hormones or enzymes, when there is the presence of this hormone, the so the certain protein cannot be accumulate in high concentrations, let's say, in certain condition where this hormone in the presence of certain hormone. So then we have to cut off. Okay, we have to cut off the accumulation of this certain protein or enzymes. So this is related to the what we call the homeostasis condition. Okay, Adriana said the overproduction of free radicals. Yeah, so yeah, betul. So oxidative damage is related to the overproduction of free radicals. So free radicals in any way it will cause the oxidation. It cause the oxidation to the proteins, to the cells. So it will cause damage to the proteins. Okay. Okay, so this is another things why we need the proteins turnover, because for the proper functions of the cells, where it requires a careful control of the levels important structural proteins, enzyme and regulatory proteins. Ini macam QC lah. Okay, so contohnya kalau let's say a certain protein is damaged, okay, it's not enough, it's not there's no presence of there is no enough protein concentration in this cell during certain times, then the cell will not function. Okay, so somehow it will give wrong signal, wrong signal to the cell. So we don't want that to happen. So that's why we need this process of protein turnover, so that somehow the protein that is damaged or not enough in a certain time, so it will be replaced with a new one, or it will be discarded. So and the degradation of the degradation generates free amino acids for protein synthesis and to fill up the metabolic process. So this is also very important. That's why we have the protein turnover, so that we can have such a process where we can actually degrade the free amino acids that no longer needed. Amount, just to show you the idea of if you look at here the amount of the protein, the protein that it's actually the turnover that happened in the high, because we know that the hemoglobin, the half-life hemoglobin pun panjang juga kan, panjang juga in the body. Who knows what is the half-life of the hemoglobin? What is the half-life of hemoglobin? Okay, half-life of hemoglobin dalam berapa hari? 20 days kan? Okay, dalam, yes, 21, okay, 21. Okay. Lepas Google lah ni semua ni. Okay, so that's why bila doktor kan, bila doktor ni there's someone pergi demam darah, okay, you do your you donate your blood, and then next time bila doktor akan bagi arahan bila you have to come back, okay, for the next bila masih sesuai lah untuk you come back untuk donate the blood, mungkin dalam masa 3 bulan lepas tu, okay, sebab the half life of the hemoglobin ni agak lama juga, 21 hari, but then the lifetime hemoglobin ni ada 120 hari, so dalam apa hari dalam 3-4 bulan jugalah. So that's why once you derma darah, okay, so it needs time to keep replacing all the blood cells semua tu, and it needs so after that kan it needs another macam 3 bulan pula lah, so you can donate your blood, okay. So that is the synthesis of hemoglobin, okay. The half-life is 21 days, 21 or 20 days. Okay. Okay, so this is the again just to show you the turnover rates of enzymes in rat liver. This is just the examples that taken from the liver of the rat itself, just only in the liver. So you can imagine in the liver too, there are presence of many enzymes. So these are the enzymes that presence in the liver of the rat liver. So here is the the half life. Okay, the half life of certain enzyme ada yang just 11 minutes, 20, 72, okay, 5 hours, even days, 16 days. Yeah? So these are so they have a different half life. And this is the KD is the turnover rate, turnover rate per day, turnover rate for each of the enzyme. Okay. Okay, so if you look at here, if the half-life is longer, then the turnover rate is much more smaller lah. Okay, much more smaller because because the half-life is just within 16 days, so meaning that it will need another 16 days for to be replaced. So the turnover rate pun akan jadi much slower lah in per daily, per day. Okay. So the thing is with the proteins or enzyme. Okay. So all proteins within cells have a certain half-life. Okay. So macam kita tengok ni lah. Okay. We see the enzymes in the rat liver pun different enzymes, so it have a different half-life. So what is half-life ni? Bila kita bercakap tentang half-life, when we talk about half-life, okay, what can you understand about the half-life itself? Okay, Okay, bila sampai masa, it means that okay, a time in which 50, bila sampai certain masa, certain time, just only 50% of the protein that was synthesized at a particular time will have been degraded. Okay, maksudnya bila sampai masa, katakan contohnya, for example, this ornithine decarboxylase. Okay, from start from so when bila sampai 11 minit, okay, when it reach 11 minutes after the first time it is created, is produced, then bila sampai 11 minutes, okay, half of this protein akan degraded, okay, bila sampai 11 minit. Okay, katakan for example ornithine decarboxylase ni, it is produced about in a concentration of 10 milligram. Okay, 10 milligram per mil, katakan 10 milligram per mil. Okay, that is the at 1 minute. Okay, so bila it is released in the cell at 1 minute, so it is 10 milligram per mil. And when it reach 11 minutes, meaning that 50% from 10 milligram tu maksudnya 5 milligram lah. So it such amount of 5 milligram will be degraded at the minute 11. So after 11 minutes, almost 50% sahaja yang akan tinggal. Okay, so that is the half-life of a certain protein. And the rates of protein turnover will vary enormously, so depending on the nature of the protein. So sama juga with the case of the turnover rates. So turnover rates also will vary depending on the nature of the protein. Okay. So between 1 to 2% of the total body protein is turned over daily. Okay, turnover daily, and this is depends on the individual protein, okay, with the function of the protein itself. So for example, for if let's say that we have this peptide hormones, so it has a very short half-life, it's just in a minute, okay, hormone. And for the regulatory proteins, let's say for digestive enzymes, RNA polymerase, this one, they also have a short half-life, which is minutes to hours. Okay, and the rate varies for individual proteins with the function of the protein juga. It depends on the function of the proteins. Katakan structural protein, which is collagen. So structural protein ni biasanya dia lebih lasak. Okay? The nature of the structural protein ni biasanya dia lebih lasak. Contohnya collagen, keratin dan sebagainya. So gonna have a long half-life compared to other proteins that have a different functions. Yeah? So this one, it can have a long half-life, even can last 4 years. Yeah? So hemoglobin has intermediate half-lives of about 120 days. Okay, and then the turnover rates depend juga on the altered demands or changes in the environment of the body. And okay, so also depends on adaptation. Okay, adaptation and juga demand. Okay, demand of the protein too, and this depends on the development, health, or illness, and also for pregnancy or lactation or other situation or other conditions. Okay, and okay, so all right. So if you look at this graph, just want you to have a wide different idea about the fractional rates of protein synthesis in a range of human tissue. So if you look at here, the different tissues of the organ, so we have stomach, esophagus, liver, colon, heart, muscles. So different parts of the organs, they have different different rates of the protein synthesis, different rate protein synthesis. So here the stomach have the highest rate of the protein synthesis. okay, compared to the other organs. So as we discussed previously pun, the rate of the turnover rate pun memang tinggi ya dalam organ dalam for the stomach. So meaning that so there will be balance lah. There will be balance ya for the protein synthesis dan juga protein degradation in the stomach and also of course in other cells as well. Okay, and we have the for cellular proteins, we have a different types of protein, which is intracellular. So proteins that will be released in the cytoplasm, we call it as intracellular. And for those, the protein that will be exported outside of the cells, then this we call it as intracellular spaces on outside of the cell, for example, intracellular spaces lumen. So this we call as extracellular protein. Yeah? And and there are two possible ways for cellular protein degradation. Okay, so the first part is the ubiquitin proteasome pathway, and the second part is the lysosome pathway. So what are the difference between this pathway? Because this pathway will provide the way we will provide approach or degradation, a method of degradation to the protein. Okay, so but there are two different pathways that can be involved, yeah, for the protein degradation. So so for ubiquitin proteasome pathway or UPP, okay, in short we call it as UPP. So UPP ni biasanya it will degrade abnormal protein and short-lived cytosolic proteins. Okay, cytosolic proteins. And so what is the cytosolic protein? Cytosolic protein. So we go back here. So it is a type of intracellular lah sebab because cytosolic protein ni a type of protein that is released in cytoplasm. We call it cytosolic protein. So it's a type of intracellular protein. So intracellular protein ni biasanya it will be degraded via this UPP pathway lah. Okay, so degrades abnormal protein and short-lived cytosolic proteins. And this ATP dependent. Okay, UPP ni ATP dependent, perlukan energy, ATP dependent. So some of the ATP, some of the energy need to be invested. Okay, need to be invested during this UPP process. It just it's not just just happen like that. Okay, without having to invest some energy. Okay, so they need the ATP, and it operating in the cytosol. Okay, UPP ni berlaku dalam cytosol. And it used two types of protein. We call it ubiquitin and proteasome. So that's why the name of this pathway is ubiquitin proteasome, because we use these two main proteins to degrade other proteins, other proteins. So ubiquitin and proteasome is boleh kata ditakuti oleh semua protein-protein, warga-warga protein dan enzim yang lain dalam badan. Okay, sebab ubiquitin and proteasome macam dia akan tangkap, okay, it will catch you if you look like abnormal or dah nak pecin ke apa. Okay, so ubiquitin and proteasome is going to catch you. It has a certain kita kata a certain mechanism yang ubiquitin ni bila dia tengok certain protein tu dia tahu okay, this is your time, okay, I'm going to pick you and I'm going to destroy you. Okay, so it has a certain ability to determine whether a certain protein need to be degraded or not. So that is very important characteristic or properties of the ubiquitin and proteasome. Yeah. Okay, so what is actually ubiquitin ni? Why ubiquitin is such very powerful? Okay, so ubiquitin ni are just very powerful, but it's actually just a small basic protein. It just has a 76 amino acids. So 76 amino acid we can consider it as a very small protein lah. Okay, because 76 amino acids is equal to just 8.5 kilodalton. Yeah, and it is widely distributed in the eukaryotic cells and highly conserved among eukaryotes. Yeah, it's highly conserved among eukaryotes, so that's why ubiquitin ni di apa it's very important, very important protein, but it is highly conserved. And this protein, when it's covalently attached to a target protein, that protein is actually marked for breakdown, so death mark. So sangat bahayalah ubiquitin ni, dia memang powerful dan macam jatuhkan hukuman mati macam tu lah. Okay. So ubiquitination ni macam a tagging system, so it has the ability to know whether a certain protein needs to be breakdown, so dia akan target protein tu. Okay, it will make a covalently attached to the protein, and then akan bawa untuk dimusnahkan. Okay. And so this is the ubiquitin structure. Okay, so this is ubiquitin structure. So it has the zeta minus, of course, this is the zeta amino, and it has glycine residue, and that attached to the lysine residues on the target protein. So ubiquitin ni dia ada glycine, dia ada glycine, this amino acid glycine in the sequence at the part of the zeta amino. So this is glycine, okay, yang warna yang black bits ni. Okay, so this is the glycine residue. So this glycine yang akan attach to the target proteins. So what happen is the glycine dekat C minus ni dia akan cari lysine residue dekat target protein, and it will attach at that location and bring or and do some rearrangement and so on to the target protein once they attach to the lysine residues. And okay, so so that is ubiquitin. So the next one is proteasome. Okay, proteasome ni apa benda pula? Okay, proteasome ni what is the proteasome pula? So proteasome ni a very large protease complex, specifically degrades ubiquitinated proteins. So maksudnya proteasome dengan ubiquitin ni is a different protein. Is a two different things. Okay, but then they work together. So meaning that ubiquitin ni bila dia mark a certain target protein yang nak dimusnahkan tu, okay, dia akan bawa kepada proteasome. Dia akan bawa pada proteasome atau protease complex ni untuk dimusnahkan. Maksudnya dia akan tangkap, dia akan bawa pergi mana? Pergi ke pusat pemusnahan macam tu. Okay. So it has two major domains, which is 20S core and 19S cap. Okay, and proteolysis occurs at the 20S domain. Ubiquitin recognition occurs at 19S domain. So it has two major domains. Macam ni. Okay, it has two major domain. This is proteasome, a very large complex protease, protease or enzyme. So it has two caps, ada bawah dan atas, and the part of the targeted protein that will be carried by the ubiquitin. So it will go through here, the cap, and it will be processed or it will be degraded in this location, proteasome. This is the part of the 20S core. Okay, 20S core, and this one is a 19S core, and this one is another 19S core. Okay. So this is how the things work. So this is the ubiquitin. So this is the ubiquitin. So ubiquitin ni dia akan bawa so this is the ubiquitin. It's very small protein. So dia akan the protein ni dia akan tag the targeted protein. So this is the targeted protein. So what happen is ubiquitin ni dia akan tagging, dia akan tag the protein, and bring it to this 90S cap of the proteasome. Okay, and akan masukkan dalam ni lah, ke dalam mesin ni, macam mesin. And then in the presence of the ATP and pepidases, so this protein tadi yang which is targeted pada ubiquitin chain tadi akan masuk ke dalam dan akan dimusnahkan dah, akan by in the presence of the peptidases. So peptidases ni dia akan cut this protein into a smaller pieces macam ni. Okay, to 25 residue for example. And this 20 to 25 residues right now, it's actually no longer in a chain lah sebab dah di-chop kan. So it will become a normal amino acid. So each of these yang smaller rod ni, and this is represents the free amino acid which is yang go through the peptidases. So this amino acid then will go to the amino acid pool back to the amino acid pool, or it become what we call as antigenic peptides. Okay? It can form also antigenic peptides. Okay. So this is in the mechanism of action in details. Okay, sekarang dah 4:30 dah pun. So I think I will just leave it. I will just end here dulu. Okay, for today. So we're going to discuss in details the mechanism of action of the UPP and also another types of the lysosomal pathway later in the next class lah on Monday. Okay. But I think maybe perhaps since I already put these notes in the putra class, so in the meantime, before Monday, perhaps you can study a bit lah about this UPP. Okay. So we have more to discuss actually, but not enough time for today. Then now we're going to continue this on the next Monday, and perhaps on the next Monday also mungkin kita ada quiz sikit. Okay, so get ready lah dengan tajuk ni, and we also going to look at the new topic juga on the Monday. Okay, so I guess that's all for today. Do you have any question before I end the session? Ada apa-apa soalan tak? Anything that you don't understand ke?
Doctor.
Yes. Who's speaking?
Fong Ying. Fong Ying speaking.
Siapa?
Fong Ying.
Okay.
Actually, I'm not really understand about the pool expansion and also the contraction.
Okay. Yang tu dekat mana dalam tajuk ni juga tadi kan? Okay, yang ni. Okay, pool expansion ya. Okay. Okay, which part that you do not understand? Sebab kalau bila say pool expansion ni maksudnya the amino acids pool itself will expand lah, because there will be increase of the synthesis of these amino acids kan. So increase of synthesis of amino acid ni it can come from the macam process of the UPP tadi. Okay, because UPP tadi akan contribute to the increase of the content or concentration of amino acid dalam amino acid pool. Okay, and at the same time there will be a transcriptome regulated. Okay, transcriptome ni adalah yang ni lah, yang whatever the protein that will be transcripted, dia akan diproses menjadi protein. Okay, benda tu berlaku, tetapi there will be unchanged in the degradation. So maksudnya kat sini semua the formation of amino acid, formation of the protein, formation of amino acid tu keep going. Okay, keep going. But then there will be unchanged in the degradation. Something happen. Perhaps mungkin the metabolism is not so active due to mungkin ada orang tu sakit ke apa, dia sakit ke dia metabolism a little bit slow, so there will be unchanged in the degradation of these amino acids. So that's why it will cause the pool expansion. Yeah, so maksudnya amino acid tu akan accumulate, dia akan expand because of this increase of the synthesis, but then unchanged in the degradation. And pool expansion ni boleh jadi boleh berlaku juga in another situation where there will be unchanged synthesis. The synthesis ni akan, the synthesis of amino acids or protein tu will be unchanged. There will be no process of synthesis of protein or amino acid. Okay, but then there will be decrease of degradation. Okay, kat sini dia unchange of the synthesis. Okay, dia tak berlaku sebarang synthesis, tapi decrease in the degradation. So dekat sini pun akan menyumbang kepada pool expansion juga. Okay, faham tak?
Ya, faham doktor. Kalau macam the factor of the pool expansion is like someone is getting ill, or is it like that?
Yeah, betul. So something abnormal happen lah. Something abnormal happen.
Okay, so for the pool contraction also the same thing lah.
Betul tu.
Okay.
So it's in a reverse way lah. In a reverse way.
Alright. Thank you, doctor.
Alright. So anything else?
No for me.
Okay. Tak ada lagi. Kalau if no, and it's already about 4:30, then I will end the session today lah. And please don't forget to go to the 12 and sign your attendance from there, from the link that are created for today's session. Okay, don't forget otherwise nanti your attendance nanti kosong. Okay, so that's all for today. I'm going to meet you inshallah face to face on a Monday. Hopefully quiz next week. Does the quiz have marks? Quiz ni biasanya it depends. It depends. Sometimes apa yang berlaku sebenarnya, okay lah, I cerita sikit lah apa yang berlaku. Okay, sometimes I buat quiz. Okay, I buat quiz dan mungkin some of the students anggap quiz or my assignment tu tak penting. Some of them dia tak hantar ataupun buat sambil lewa je the quiz, the assignment. Okay, the thing is bila final marks, okay, bila you get your final marks, or I think that okay, perhaps your marks dekat borderline, macam mana I nak tolong? Okay, so this is very important. Quiz, assignment, your lab report is very important. Bila I tengok your marks dekat borderline, macam mana I nak tolong? So I go back to your quiz, I go back to your assignment, I buka balik dekat mana I akan cari, kat mana I nak tolong. Okay, so that is the thing with the quiz or assignment. So take it, don't take it lightly, anything yang kita bagi. Sebab itulah yang saya cakap tadi, case borderline ni sangat critical. Okay, but then if you deserve that marks dekat borderline, then I will just leave it there lah. I takkan buat apa-apa lah, even though you dekat borderline, sebab I dah consider semua, your quiz, your assignment, and I cannot change anywhere, then that's it. That you deserve it lah. That borderline marks. Okay. So anyway, itu my pesanan lah. Okay. Alright. Okay, Adriana, thank you for the interesting question. Alright. Okay, so I guess that's all for today. So thank you, yeah. So don't worry, don't forget the attendance. Okay, I'm going to end the session. Bye. Stay safe, everyone.
Thank you,
Thank you,
Doctor.
Doctor.
Thank you, Doctor.
Thank
Thank you, Doctor.
you.